Okay, so my name is Dana Zoldan, and I will show you today all the tips and tricks that we have in Phenom that will help you to make your work more efficient and easy. So let's start. Before we will start, let me go over. Okay. We go over a little bit about the Zoom. For the one that doesn't know, we are going to have around 45 minutes of presentation and some live demo. And we will have 15 minutes for questions if we will have some. During the session, you will be able to ask questions to our panelists through the Q&A. Our panelists today will be Ruben, Yael, Mariana, and Tamar. They will be happy to assist you with any question that you will have regarding the subject. You can also raise a hand if you have any issue. And please just ask questions through the Q&A, and this way we will be able to monitor them and to answer all of them. Okay, so for today's topic, we have some column management tips and tricks. Field related, we're going to talk about the index function and sub-level calculations, and also some general tips and tricks. Let's start with some column management related shortcuts, and I'm going to start with the variables. So we know that the variables are important topics in Phenom, and they can represent traits, factors, codes, name, anything that will relate to the germplasm itself. In Phenom, we have more than one level of variables. We have the germplasm level, which with the green leaf. It's a level that is related to the germplasm itself and will be shown in every field, any table, any grid that you will show this variable with this icon. Then we have the field level variables, which are in the list tab, plot tab, and here we can see the observation tab with the magnifying glass. And also we will have, in this case, the inventory. Same variable can be presented in different levels, depend on what we want to show. For example, here we are showing the comments. A little bit about the difference between germplasm level and field level, because we know that this is an important issue that some of our customers are asking a lot about. So, as I said, germplasm level information are the data, the passport data that we have of the line of the germplasm, for example, name, resistances, viruses, generations, and so on. And then we'll have the data that we are collecting in the field. For example, measurement of germination, height, and so on. So the difference between those is that the germplasm level information will be shown in every grid and will not change during observations. And here I can show an example that with each observation, we will have a different measurement, and not all of them are representing the germplasm. If we will find one measurement that is representing the germplasm and we would like it to be in the plant, in this case, plant height, in the germplasm level info, we will have the option to copy that, and then it will be displayed in the germplasm level information. I will talk about that later, and I will show in details how to do that. More a little bit about column management. So what we have is that we can define with the columns to show what germplasm columns will be displayed in a grid. So this is available for all the navigation panel objects, and we can access that from actions and then define germplasm columns. Then we have another level of the columns to show, and there we can choose to hide, display, or reorder the columns in the grid. This is available in all grid, and we are doing that from the toolbar itself, from the grid itself, clicking on the icon of columns to show. The last thing that we have is to add column. Add column is to add columns at the field level, columns that will be with the field column icons. This is access in the field. We have access for that from the field tabs. and we are clicking on the right action button, add column. Define germplasm columns. We're going to talk about that for a few minutes. So we're doing that from the navigation panel on the left. We're selecting the entity that we want to add those columns to. We're dragging the wanted columns from the available to the selected and clicking on OK. With this option, with the define germplasm column, we'll also be able to add female and male data, previous generation data, for any germplasm column that is displayed. How do we do it? We are dragging it from available to the selected. We can see in this case that we have 3 columns of breeder name. Once clicking on the type, double click on the self, which open a drop-down menu, and we will be able then to select the wanted female or male. So in this case, once we are adding the breeder name, we will also be adding the name column. The name column of the female or male cannot be added alone, only with another column because it's a software column. So once I will add the breeder name, I will also add the female or male column. So this is how it looks like once we are adding them. Female and male columns will have the female and male sign, pink for female, blue for male. And then we can see here that we have both the name and the generation display. This is a very useful feature that will allow you to display the columns of the previous generation and the data. So it can be used in every grid and it can be shown in all of the columns that we have on the germplasm level. Make sure that you are not trying to add columns that are, for example, a field column that is a system column, and then it can stack the germplasm columns and it will not be displayed. Columns to show. So with columns to show, we are able to arrange and hide columns that we don't want to see on the grid itself. So we can have several columns that we don't want to see, then we can hide them. We don't need to delete them, we can just hide them, we can rearrange the order, and then clicking on OK, and it will be displayed then. We can also add a column on the grid itself, on the field level, by clicking on Actions, Add Column, select the wanted columns either from a label or one by one by searching with the filter, and dragging them down. It's important to drag them down, make sure that they are in the selected variable. Then clicking on OK, and they will be added with the icon of the tab they belong to. So this is the icon of the magnifying glass in the observation, which means that I can only add this column at the observation level. So if I will see plot level column at the observation level, I won't be able to edit that. Just the columns that have the same icon. Okay, so here are some shortcuts that we have on those. I will talk about the column template, but know that we have also the variable labels and the copy column configurations. So for columns template, we can save a set of columns template for any grid in Phenom. Each grid has its own template. So for example, if I have a template for plot, then I won't be able to use it at the observation level, but I will be able to use it in another field in the plot level. So those sets of columns can be later loaded, and it will save you a lot of time when searching and changing columns. Variable labels are an easy way to group traits and add them to the field easily. It's available in all the field grids, and it is done from the variable level. So later I will show you how to access our knowledge base, and there you can also search for all of the things that I've explained or things that I've mentioned and didn't go in depth to them. The last column management shortcut in this case is copy columns configurations between fields. And what this option does, it can copy the entire configuration of the field for all of the grids. So for example, if I'm copying a breeding field, then I will be copying the list, the plots, the observations, selections, and crosses. So all of the tabs that have configuration for columns will be copied. Let's talk about the column template. So this is a really useful feature for us because it can save us a lot of time. So for example, if I want to display and to load an existing column, I will click on the columns template icon. Then I will click on load and choose the wanted template. If I want to create a new one, then first I will have to arrange all of the columns in the field itself. Then click on the columns template and save. Later, I will be able to load that in any other, in this case, if I'm doing it on plot, in any other plot level in the fields. Now we're going to some, if we talked about the field, and we're moving to the field again, and field-related actions. We will start with clone fields. So clone field will allow you to duplicate field, including all of the settings and the content, the list, the map, the plots. We can clone up to 100 fields. And this is useful in case that the user wants to have the same experiment with the same germplasm, the same map, the same plot name, just in different locations. So once I'm clicking on the field name, then actions clone field, I will be asked to choose how many clones do I want to create. Refresh the browser tab, and then we will see that we have here one's observation and 4 copies of that. Then in order to rename it, I will stand on the field itself and actions edit, and then I will be able to edit the field's name. Another field related setting is field default variables. And here we can define field default variables on the field level. So it will not appear in any of the tab, it will appear as metadata of the field. Things that is defined for the entire field, such as treatment, watering, a year, location, those kind of things that are important for you to represent. So we can have it either as default by adding it from the setting, or we can have it by clicking on the variables when creating the fields and add them. Once we are adding them as default, we will have to fill them in order to create the field. Those variables that we are adding as metadata to the field will be also can use them in the naming convention later. When creating selection or process, we can use this data such as year, such as location to add into the new germplasm name or any other column that we would like to add. Replace germplasm. So this is also one of the useful options that we have. So this is in case that you have sowed a germplasm, you have created plots from it, and also created future selections. And then you're going to the warehouse and you're finding out that you don't have either enough seeds or seeds at all, and you need to change the germplasm. So what do? Instead of deleting selection, process, observations, we can replace the germplasm, and it will change germplasm in each one of the levels that we have on the field. So this is done from the list level, and we are selecting the germplasm that we would like to replace. Click on action, replace germplasm. Then we will search for a germplasm that we would like to replace it with. Click on it, and the germplasm will be changed in all of the levels in the field. Copy to germplasm. This function will allow you to copy germplasm field data to the germplasm data. So what it means? It means that, as I showed in the previous slide, that if I'm checking height, and the last height that was measured is the height that I would like to keep, a height that I feel that is representing the line, and I would like to see the germplasm, and I would like to see it at the germplasm level. So how is it done? It is done from the field. I will from the observation level maybe most of the time, but can also be done from the plant level, from the plot, and from the list. I will select the germplasm that I would like the column to be copied. Click on Actions and Copy to Germplasm Level. Then I will select the wanted variables. Make sure that you are checking the black V and not just selecting the variables, because if you will just select the variable and not click on the black V, it won't happen. Checking the black V, clicking on OK, refreshing, and then seeing that we have a new column that is called food shape, and the data was copied and now is displayed in the germplasm level. Okay, so recalculate. Also an important feature that will allow you, if you have made changes in the parent's data or in resistant data of the parent, either general data of the parents or resistant data, and it is done after creating already crosses and selection, we can use this to refresh and to recalculate the inheritance rules and the naming convention rules in order to take the new changes and to display them. So how is it done? I'm selecting the field name, clicking Actions, and Recalculate Crosses and Selection. Again, I will choose either selection or crosses in the type. Make sure to check the black V. You can select both selections and crosses. Then I will choose the variable type, either inheritance rules or naming convention, and I can choose both. And last, if I would like to override values that have entered manually, I will check the override box. If I don't want it, if I have made changes and I don't put in them manually, and I want the recalculation to be done only on things that I haven't changed manually, I will keep this box unchecked. Another important regarding the recalculate, make sure that in the naming convention settings, the inheritance box is checked. If you don't know how to check it, please contact your key user or Phenom in Phenom open a case. Okay, so now we are moving to the index function and sub-level calculation. So those are arithmetics and index function that will allow you to do a lot of things in the field level, both in the selection level, observation level, plant level. So how is it done? So here I'm showing on a plant level, for example, a bunch of resistance that we're taking, and here I'm seeing it only as R and S, and I will have to look at the column name in order to know to which is resistant and what has the resistance in it. So what do I want to do? I want to have an already visualized column to show me the resistances. So how do I bring it? I will add this column on the same level. So if I'm here on the plant level, then the column will be on the plant level also. I will click on Action and then Set Value. Then I will choose the column, in this case, resistant 2. Then on the 2, I will choose the index and I will start to play. In this case, I'm choosing the concatenation, but I can also do a sum or mean. Then I will choose the first variable that I would like to take in consideration, in this case, I2. I will enter a condition and the result of it. So in this case, because I'm doing concatenation, then I would like to have an alphabetical value representing the column. So here in this case, I'm adding an I2. I will add it to each one of the different columns. I will have add variable, and then I will add to each one of them. Then I will click on OK, and it will look like that. So I will have resistance 2 and all of the resistance that I have for this specific plant. Then also important that this data that we have on the plant level can also be copied if I'm creating selections from it. So if I'm copying selection and I want it to be displayed, I can choose display it from the plant level, and then I will know what exactly the resistance that I have for this specific plant selection later. Another very useful feature is the sub-level calculation, and for this one I will also show a short demo because it has a lot of stages and I would like to go step by step. So this function is allow you to run calculation on variable, but not as columns as I showed you with the index, but for different observations. So how do we do that? First, we will create the column that we want the data to be gathered on. In this case, we're using maximum germination. Then I will select it on the variable level, click on action, and then function. It is highly recommended to do it from the research group level in order to see all of the variables that you have. Then I will start to fill in the form. So I'm starting with the source variable. So what is the variable that I want the data to be taken from? So in this case, the source variable will be germination. And then I will choose the function that I would like. So in this case, I would like max to see the maximum germination that I have. I also have min, sum, count, minimum, and first result that was recorded, and last. Then I will choose the main level. So the main level is the level that I want the data to be shown. Okay, so if the data was collected on the observation, then I want to be displayed from where the observation was created from. So if I created the observation from the list, or from the plants, or from the plot, so this is where will be my main level. The last thing that I will do in order to show, to finish this function, is to choose the sub-level from where the data will be taken from, where the data was collected. So usually it will be in observation, but in some cases it can also be in selections or in plant. Next steps will be to add the variable to the field in the wanted level. So if the main level is the list or the plot, then I will display there the column of maximum germination. And then also I will make sure that the column of sub-level in the observation will have the germination column also. Okay, so here I have 3 germination results that was collected in different times, and I want to show the maximum germination. So how do I do it? First, see that I have the germination percentage column already here, and also the maximum germination. And you can see that those are in different levels because the germination have the magnifying glass icon, which means that it is under the observation tab, and the maximum germination has the plot, which means that the origin of this column is from the plot, and I can only edit that from the plot. Then I will go to the field level, select the field name, click on Calculate Variables, and then the data will appear on the plot level. I want to show you that also, sorry, on the demo itself. Okay, so here I'm selecting the field, but first I will go to the tomato, we'll choose the variable. I have the maximum germination, clicking on it, action, functions, and here it's already set. So the source variable, as we said, will be the germination, the function will be max, the main level will be plot, and the sub level will be observation. I can choose more than one level if I would like to. I don't have to choose just one. After I have created the function, I can go to the field, make sure that I have the data on the plot level, and that I have here 3 observations. So here it's already been calculating. Let's clear that. Go. And then I can go again to the breeding, action, and recalculate, calculate variables. And the data will appear again. Another thing that is very useful regarding this feature is if I will use one value, if I will change one value, for example, if I will change the 87 to 93 and the 89 to 78. Okay, you can see that I changed them, they're highlighted in blue. I will refresh in order for them to beat the system. Once I will refresh and I have function on this field, I'm having this yellow banner. The data was changed, and to calculate functions, click here. In order for us not to go all the time back to the field and click on calculate variable. So I will do it again, just so I will see the banner, clicking here, and then the new data has changed for here for 93 and here for 78. Let's go back to the presentation. Okay. So the next one, which is also very nice feature, is the arithmetic function. And with the arithmetic function, we can add, subtract, multiply, do a lot of field calculation for different columns. So in this case, I'm using to do a sum. I want to see the total number of fruit, but I can also in some cases do an average, or I can multiply and divide and create, let's say, for example, yield per hectare or yield for plot. Okay, this is also can be done. And let's see how it's done. So I'm clicking on action and then set value. Then I'm selecting in one the column that I would like the data to be presented in. So in this case, all of the columns are at the same level. So make sure that all of the columns will be in the same level. Here we are doing it on the observation level. Next is to build the function. Choose the first variable, click on the green arrow, add a operator, and then continue to do it until you will get to the end of the function, to do what you want, clicking on OK, and the data will appear. I will also want to show you that on the main software. Okay, so here I'm having open field and I have all of the data already. What I want to show you here, set value, choosing the wanted column, then I will go to function, and I already have here the function, but what I can do in those cases, and this is thing that we haven't had in the past, we can also use date. So for example, if I'm entering a date column, it will also be calculated as a numerical column. So this is the total number of fruit, and on to the next step. Okay, so now we are going to the more general tips and tricks. So the first one that we will talk about today is the inherit. So it's also a very important action to do when you are changing data from a parent to an offspring, and you want to change it for the entire offspring. You don't want to change it just in one field. If we would like to change this in one field, we will use the recalculate like we did before. But here we want to change it for one germplasm, but to all of its offspring. So we are doing it from the inherit, and this function will work on the variable that has inheritance rules or naming convention. If I don't have inheritance rule or naming convention, then it won't work. If you want to have inheritance rules and naming convention, please contact us at openacase@phenomenetworks.com. I'm clicking on actions and then inherit. Then I will be asked to choose the variable. And then again, if I would wish to override data that was entered manually, I will click on it. If I know that the data that was entered manually is correct, then I won't click it. Then I will click on it. Then I will choose if I want it to be on selection or process or on both. And this will be done for the entire pedigree for this line, from this line onward, which means that if I have selections and selections of selections, it will also be copied to that. For crosses, it will also be copied to that. So please use this feature very carefully because it will overwrite data and it can change all of the pedigree along. So make sure that you are using it with full confidence. The entities tab. So this is one useful tip that I can give you. For all of our customers that have a lot of data, a lot of entities, a lot of fields, and you don't want to do all of the clicks, go to the entities tab on the higher level, enter the field name or field code, and then click on the blue box, and it will open a new window that will set you directly to the field that you would like to go to. So you don't have to do a lot of clicks, just search the field, click on the blue box, you will be right there. Another very nice feature is color by input. And what this feature does, it will show you what is the source of the data, how it was taken. Was it taken manually? Was it uploaded by a file? Was it created by inheritance rules? So this feature, we're highly recommended to do it on a folder base and not on the entire research group in order to not to slow the system. So if you're doing it on a specific folder, then it will be just on that and it will not affect the entire software, the entire database. So what do we see here? Variable that was created when the system applies naming convention or inheritance rules, which mean that they were created by the system. We will see this most on selection and crosses. They will be colored in green. Values that were uploaded from an Excel sheet will be colored in blue, and value that was inserted manually will be colored in red. Value that was created on selections and when entered with our phenotype will be considered entered manually, so they will also be colored in red. We'll also see some columns that are black. With black here, you can see that those are black color, which means that those are software columns. They will be the GID, name, method, cultivar, and field column will be colored in black. How is it done? How do we get to these settings? We are going to the settings level on the folder, clicking on actions, setting, scrolling down to color by input, checking the yes, and checking the yes here also. Attach a document. So we have been asked a lot to have the option to add documents, either PDF or Excel or Word document. That can be assist in case that the specific field have had some soil test or some data that you would like to upload and to be linked to the field and available to all of the users that will have this field. So how do we do it? How do we upload files? We are selecting the entity name, so we can do it for folders, field, germplasm set, list of lots, any entity, any icon that I have on the navigation panel. I will select it, click on edit, Then I will see on the left bottom corner document, I will click on it. And then I will be able to upload document. I will click on add, choose, and then I can add a short description, click on add, and then I will be able to open that. So those are existing document. I'm clicking on open, and it will download the document to the computer of the user. Okay. Another nice feature that we have, and I don't think that a lot of our users know about it, is that you can send germplasm to a specific germplasm set from a pedigree chart. So sometimes you will create a pedigree chart and we'll see, okay, so this germplasm is very interesting to me. How do I remember that I would like to use it in the future? So this is how. I will open a pedigree chart. In this case, I'm using the normal view. Then I will see all of the pedigree. Just a quick reminder, the yellow one is the one that I chose to see, the highlight one. Orange one are selections. Green one are process. Dotted lines are the males. Solid lines are the female. Then I will click on the germplasm that I would like to select. They will appear here on the selected germplasm. Click on send. Choose the germplasm set that you have created ahead, and it's done. You can later go to that, and you can continue and work with those germplasm without writing it down or just by sending them automatically by the system. Downloading file format is that you can usually, as you know, working with Phenom will require work with CSV file and not Excel files. So what we did here is we added an option to change the file that will be downloaded to the computer to be CSV instead of Excel in order to save time, and you won't have to save it as CSV. It will be downloaded as CSV already. How is it done? We're going to the germplasm, to the research group. Actions, and scrolling down, downloading file format, change it to either CSV or Excel, and it's done. Then every file that you will download from Phenom will be in this format for this specific germplasm set, a research group, or folder. Okay, data integrity. So this is a very interesting feature that will allow you to check if values in the grid are not matched with the variable scale. So how is it, you can ask yourself, how is it even a possibility? Because we have restriction. So this is done, and you can see it here from either data that was uploaded before we have set the scale, or data from a phenotypic study that was uploading as observation already. And how do we do it? We are selecting, we are going either to the Germplasm tab, Observation tab, or Field. Clicking on Actions and then Variable Integrity. It will highlight in orange the values that are not consistent with the scale. Okay, so we are nearly at the end, and I'm going to talk now about two very, very important features that are in Phenom. I know that a lot of our users are using them constantly, but I think they're important enough to show again. So we have the sorting option, and here what we can do with that, we can sort, and we can sort not just on one level, but on several levels. So I can sort, for example, by first I will start with generation, then I can go with the type, and then for method, for example. Or if I'm at the field, I can sort by plot name, then by replication, then by origin. I can do a lot of different layering for this sorting. So how is it done? I will select the sort on our toolbar. Then I will drag the wanted column from columns to sort columns. Then I can leave the sort automatically, or the recommended option is to choose whether it's numeric or case sensitive. Then I will choose if I want it ascending or descending. If I want it ascending, then I will check the no. If I want it to be descending, I will double click on the no, and it will change to yes, and it will show first the higher results or the latest results and the smallest results at the end, or if it's alphabetical, then from Z to A. Okay, one last thing that I'm going to talk about, the filtering. So I know that a lot of you are using the filtering, but here are all of the options that we can use to use the filtering that I know that some of you don't know that exist. So we have the numerical values that are greater than, lower than, greater than, equal. We can also search for results that only have some of the characters. So if I'm entering Z or 3, then everything that have Z with it will appear, or anything that have 3 with it will appear. If I want the exact value, for example, I just want to see the 3s and not 3, 13, 33, 23, I will add the equal to sign before the value. So then it will show me only the 3s, or only the things that have only Z within them. For range of values, I will add the 2 dots between the range. Results that contain specific numbers, I can use the comma separator to show that, and make sure that if you have an entire number like 13, 14, 50, 55, enter the entire number or the entire combination of number and letters in order for them to show. Okay, so results with empty cells that don't have any data within them, I can click on curly brackets, the word empty, and then curly brackets. And for results that have data within them, and I think this is now hiding. Okay. Then I have the not empty with curly brackets, or I can also use the percentage sign to filter only cells that have data within them. Okay, so I have finished with the presentation. Let me see if you have any questions. Okay, if we don't have any question, then we will finish this presentation. Let's give it 2 more minutes. Yes. Okay, so let's start. Okay, so how to set the field data templates, which means that if I would like to set the field data templates, this is done from either I can choose a field that will be the template, and then I can copy from there with copy columns. So this is how I can do that. I can also do a different templates for all of the different levels, and then I can copy from the template itself, from the column templates. No, Magda, we haven't talked about the cultivars. If you want any answer regarding the cultivar, if you have a specific question, please email that to openacase@phenomnetwork.com, and we'll be happy to assist you. Are there any configurations that can make the female and male variables invisible? We will have them in the future. We will have the option to hide the female and male. For the time being, what you can do, you can use the permissions and then prevent a user from changing the columns. So he will be only able to see the columns that he has, and not and not will be able to change it and or to add any columns. If you want to know about specific cases or to create a specific permission, please contact us as phenomnet at openacase@phenomnetworks.com. Let's see. How to find the germplasm columns in Phenotop while taking the same field observation. I'm sorry, please send this question to openacase.com. It's not relevant to this webinar. How to modify, okay, so this is a good question. So column templates cannot be modified manually. What needs to be done is if I have created a template and I would like to change it, then I will have to click on Manage, delete the old template, and then to save it again. See on the chat. We already have a webinar on Phenotop, and you can search for that on the knowledge base. And this is a good time to show you the knowledge base. Okay, so this is Phenom knowledge base. So it is, I don't know, almost a year old. And it's, I think, one of the most helpful features and tools that we have. We can search here for everything that we have on Phenom based on keywords, or that I have an already created topics. If anyone asks about the mobile app, then clicking on the mobile app, I have here data regarding the Phenotop and the Phenom app. Just for you to let you know, we are not supporting the Phenom the Phenom app anymore. If we have any bug, we are moving that to the support, and we are recommending to start to work with the Phenotop. It has a lot of more options. It's easier to use, and the syncing is done automatically. Let me show you just for a word how it's looking. So we have now a question how to create a trial, and I guess that this question was related not to cloning a field, but to create a trial when one has used the already field design. So I create go to field, then create a field, and you can see field type, but this is very starting data. But later we can see from recent content the list, the plots, observation, the map tab, and here in the map tab we will find our answer. So we are going to map, randomization, and here toggle border and the experiment and design tab. And from here we can create the different fields. So this is not done from the field anymore, it is done from the experiment tab. If we want to create duplications, let me show up. If you want to create duplication, then it's done from the experiment. Please search for that on the knowledge base, and if you have any difficulties, contact us at openacase@phenomnetworks.com. Okay, so to copy data from one column to another column, we will use the set value. And this is done from a field. Let me see if I have perhaps here an example. Let's add just one more column. This one, just to show for the example of it. So I have two columns. And I'm clicking on Actions, Set Value, choosing the column that I want the data to be copied to, choosing Text, then choosing the column that I want the data to be copied from. So I have the germination, and I want it to be copied to the commercial, clicking on it, and then the data was changed, was copied. Okay, so if we don't have any more questions, I would like to thank you for taking the time and being with us today. We're going to have more webinars on different topics. If you have any requests, please send this to us to Open a Case, and we will try to have 1 or 2 webinars in a quarter. So we'd like to have your requests, so we'll do it according to your wishes. Thank you very much, everyone. Have a great day. See you in the next webinar.